So we're looking at a paper which is dealing with term two's work. So it starts from genetics, somewhere in the middle of genetics, and it includes all of the nervous system as well as the endocrine system. Okay, so you'll start off with the instructions. Answer all questions neatly in blue ink on the line paper provided and rule off after each question. Right? It's important that we follow those instructions. Right, then we get to the multiple choice. It says how many synapses would an impulse need to pass in order to move from the receptor to the effector in a reflex arc? Who can give us that answer? Right, Katleho says B, which is correct. Okay, so remember in a reflex arc, the impulses are generated by the receptor. They'll move from a sensory neuron between the sensory and the interneuron. There's one synapse. Then with the interneuron, when it gets to jump, it jumps a second synapse and then goes to the motor neuron before it carries on, right? So normally we're looking at 2 there. So the answer is B, correct. Then they're saying a male with hemophilia marries a female who is homozygous normal. What is the probability of them having a child with hemophilia? What's your answer there? Right, remember, if it's a male with hemophilia, how would you write the genotype for that in a sex-linked disorder or example, right? In a sex-linked cross, for a male, how would you write the genotype? Right, you guys are a bit slow today with the answers, right? For sex link, because it's linked to sex. Okay, very good. Thank you, Fatima. That for sex link, we're going to have to show X and Y, right? So a male will be XY, but having hemophilia, is having hemophilia a dominant or a recessive trait? That's one thing we should know, right, from the content that we've learned already. That to have hemophilia, remember there's 2 sex-linked disorders that are found in the exam guideline, right? One is hemophilia and the other is color blindness. So to have hemophilia to be affected, is that a dominant characteristic or is that a recessive characteristic? Because now we need to know, must we put a capital H or a small h, right? So it's recessive, thank you, Katleho. So we're going to put a small letter H, okay? And then normally you'll have that in superscript there, okay? So the Y, remember, doesn't have anything, very important in a sex-linked disorder. Good. Then they're saying he marries a female who is homozygous normal. Now, normal obviously would be then the capital letter. So a female would be XX. Remember, they've given us a homozygous normal. Normal is the phenotype. So now we need to convert that to genotype. So a female who is normal is like this here. And here, it's a non-carrier, okay? It should have maybe been more specific on that. Okay, no, sorry. They didn't have to say it's non-carrier because they said homozygous. Homo means the same. So once you've got X capital H, the other one has to also be X capital H. Right? So that's a normal female. Now, if you're going to do a cross with these two, what's the probability of getting a child with hemophilia? So here they're just talking of children, whether male or female, doesn't matter. We're just looking overall the children. Okay, so Iram says the probability of having a child with hemophilia is 100%. Let's see. If we take the male, okay, change your answer. Okay, let's see. For the male, I separated the gametes, right, and put into the table. Remember, you don't need to do a whole cross to start with the P1 phenotype, genotype, etc. Because here we're talking of specifically just a multiple choice. It's not for 6 marks where you need to do that whole format, right? So here we just make a quick table on the side. When we then start to now copy okay, it's doing that, right? But we want to take this and paste it there. From the mother, all of the children are going to get X superscript capital H, which means a normal allele. And then from the father, the daughters will get this recessive trait, and from the father, the sons will inherit the Y chromosome. So now if we look at the offspring, which are here, the female offspring are X capital H and small h. Is that affected by hemophilia? It's heterozygous, having the allele for having it, which is a small h, and the normal allele. So this one here, as Kasako is saying, that it's not having hemophilia. Why? Because the dominant allele is the normal one, which will overpower kind of the recessive one, right? Mask it, and you won't see the recessive trait. So both daughters are normal, and both sons are normal. So the probability of them having a child with hemophilia is therefore 0. There's no child that they can have with hemophilia. So the answer is A there. Okay, then they're saying insulin is secreted from which of the following? Is it A, the liver, B, all pancreatic cells, C, muscle cells, or D, the islets of Langerhans? Right, Katlako says D, and that's correct. If you look here, the distractors A and C are the target organs, right? So where the conversion of glucose to glycogen occurs. Okay, and B is close because it is the pancreas, but it's saying all pancreatic cells. Certain pancreatic cells are making pancreatic juice for an exocrine function, right? So here we can see that the correct gland from where it's secreted is the islets of Langerhans cells within the pancreas. So D is the most correct answer. Then we get to giving the correct biological terms for the following. A mutation that doesn't affect the survival of an organism. What would you call that? One that doesn't affect the survival. Right, it's harmless, good. And then a change in the sequence of nucleotides. What's a specific name there for that? When there's a change in this sequence of nucleotides. Right, very good. Humera's got mutation, which is correct, but the most correct is a gene mutation. Remember, there's two types of mutation. So a mutation would be just a change in the genotype of an organism. And then if you're talking of the sequence of nucleotides, then that's a gene mutation. Whereas if it's in the structure or number of the chromosomes, that's chromosomal aberration, the other type. Right. So the best answer here for 1.2.2 would be a gene mutation. Then 1.2.3, inserting of genetic sequences of other organisms into a crop to produce desirable characteristics. What's that called? Right. So if you insert the genetic sequence, basically a gene of another organism, into a crop, for example, right? So from one organism to another, and then you get a desirable characteristic for us. What's that process called? Biotechnology, not exactly, right? The whole section is biotechnology. Biotechnology, using living organisms for the benefit of humans, but that includes things like stem cells, right? Using living cells to benefit humans, which is not this specific example that we're talking of. So stem cells is part of that. Cloning is also part of biotechnology. But here we're specifically talking of taking a gene from one organism and putting it into another one, right? That's the beta Iram. It's genetic engineering, right? Or genetic modification. Okay, then they're saying the gland that produces ADH, antidiuretic hormone, where is it produced? What's the name of the gland? Who can help us there? The hypothalamus, Katlako, very good, right? So a common wrong answer here would be the pituitary. Remember that it's produced, ADH is produced in the hypothalamus, but released from the pituitary. So made in the hypothalamus, released in the pituitary. Okay, I'll show you the memo just now, right? For those who missed maybe one or two. 1.2.5, hormone responsible for male sexual characteristics. That's an easy one. Okay, testosterone, good. Right. Good. So if you look at the memorandum, right, the multiple choice had bad answers, and the one-word answers. Remember, multiple choice is 2 marks each, right? The one-word answers: harmless mutation, gene mutation, genetic modification, or genetic engineering. They accepted GMOs, which is genetically modified organisms here. But although it's not the organism we're talking about the process here, right? So, but genetic modification or engineering would be a better answer. And then hypothalamus 1.2.4 and 1.2.5 is testosterone. Okay, we get to the matching of columns, right? They're saying 1.3.1, the number of X-shaped chromosomes in a male. Now, this was actually a trick question, right? They're not saying the number of X chromosomes. The question is saying the number of X-shaped chromosomes. So who's got the answer there? Okay, Kataka is saying one, A only. Okay, anyone else? But most would say A only. But I was being a bit technical when I said this one, just to test you guys out, right? We're not saying the number of X chromosomes. We're saying the number of X-shaped chromosomes. So if you look at, okay, so we've got 3 different answers. Unfortunately, all are wrong. The reason is that if you look at a karyotype, a diagram showing the chromosomes of an organism, right? Now, this one here is showing 3 at number 21, which is Down syndrome, right? So it's not a normal situation. We're talking of a normal human being. A male will have 46 chromosomes, 23 pairs. Each pair is a homologous pair, meaning the same size, the same shape, same centromere position, same genes. But the last one, the gonosomes, the sex chromosomes, is going to have an X chromosome, which is a larger one, and the Y chromosome, which is a smaller one. But this question is not talking of X and Y chromosome, it's saying the shape. So if you look at all of them are made up of 2 chromatids joined together a centromere. And that makes an X shape. So all 46 of these are actually X-shaped. So the answer that I wanted there was to say that none of them. It's not 1, it's not 45, it should be 46. Okay, then we say, can be used in paternity testing. Can blood groups be used in paternity testing? Yes or no? Right, when you're answering this, you probably say, okay, it's not 1, and this one wasn't 45, and then you say, therefore, it's none of those, right? Can you use, good, right? So we've got the answer there that it's both A and B, right? So blood groups, yes, we can use them for finding out paternity. Remember, there's certain options that you can't get. If both parents are maybe type A, B, then, and the child is type O, then you can say, is this the father possibly? It's impossible that, right? So you can use it to kind of rule out certain parents. You won't say definitely this is the father, but it can be used in that process. And then DNA profiling, most definitely. So the answer is yes for both. So the answer, therefore, would be both A and B. Then it says secretes 2 or more hormones involved in reproduction. Does the ovary secrete 2 or more hormones in reproduction? Right? Humera says both. It's good. Correct that. Who can tell us the ovary, which hormones does it secrete in reproduction that are required for reproduction? But remember this, even though it's talking of reproduction, it's still repeated this information in the section on the endocrine system, right? So correct, Karodia, to say that estrogen and progesterone are secreted here, right? And then from the pituitary gland, which 2 are secreted that are involved in this process? Pituitary gland secretes 2 hormones also that are involved in reproduction. That's good, LH and FSH. Good, right? So those 2 mean that both A and B satisfy the statement in column 1, and therefore the answer is both A and B. So if you look at our answer there, the first one was none, and then both A and B, and both A and B. Okay, if we move to question 2. Study the diagram and the graph below and answer the questions that follow. So they've got a diagram. First thing we should do is label everything, right? So what is the label for A? And what's the label for B? And what's the label for C? Basically, let's see if you can give us those answers, please. Right, so A is the iris, correct? B is the retina, which is correct, the innermost layer, and then C, yellow spot, perfect. Right, there is another name, the fovea centralis, but yellow spot is accepted in the exam, so that's fine. Okay, then if we look at the graph, we can see it's showing you the relative clarity of the image. How clear is the image? It starts off low, then at 1 it's slightly higher, then goes down to nothing at 2, and then it increases a lot at number 3, and then it decreases after that. So they're saying explain one way in which part B is suitable for its function. Part B, the retina. How is the retina suited for its function, right? So when you talk of suitability and it's explained, so you need to give a statement and a reason. Suitability basically means structure and function. What structure does it have? And then how does that help it in fulfilling its function? Okay, good. Katleko says it has photoreceptors, good, which are the rods and cones, right? So would that give you 2 marks? No, right? That's the first part. You're saying it's got rods and cones, or it's got photoreceptors. That's fine, 1 mark. Explain, right? And good, Iram goes further to say, receive stimuli and convert them to impulse. And we've got that answer coming again from someone else. So that's good, right? So that is the correct answer. And then 2.2. Explain how and why the part labeled A makes adjustments when a person moves from bright light to dark light. Okay, so now we're talking of changing from bright light to dark light. So how does it make the adjustments, and why does it make those adjustments? Explain. So there'll be a reason with it, a statements and reasons. As you can see, it's for 5 marks here. So we can, instead of trying to type maybe all 5 points, type 1 and then you can send through something good. So it's saying that the circular muscles relax. Good. If you followed the videos and what we've done in class, we said that when you're looking at dull light, then we find that it's CR and RC. Good, right? So the radial muscles of the iris contract and the circular muscles of the iris relax. Very good, right? It's good. I like that all of you all have named, not just say circular muscles, say of the iris. That's good. And as a result of that, the pupil will dilate. Very good. Okay, so that's how it happened. Why must the pupil dilate? Now that's the next part of it in explaining, and that's how you're going to get your 5 marks. So far, what we've said, you should get about 3 marks for it. Right, good. So more light enters the eye. And why do you want more light to enter the eye? Very good, Humera, to say, for clearer vision. Okay, and then the last question. Okay, there is still more, right? Let's just check the memo to make sure we are all on the same page, right? It has photoreceptors or rods and cones, 1 mark for that. And that converts the light stimulus into an impulse. Okay, so we said stimulus to impulse. It would be better to say the light stimulus because then you're specific to the eye now. Then the iris contains 2 sets of muscles. We never have that point, but the radial muscles will contract, the circular muscles will relax, causing the pupil to dilate, which allows more light to enter and to form a clear image. So we've got all of the parts of that. It says any 5 of these 6, so you would have got full marks for what we had there. Then they're saying which number 1 to 4 on the graph represents C in the diagram. C is the yellow spot. So which number here represents the yellow spot in the graph? Number 3 is correct. Good. And then the next question is saying give a reason for your answer. Why would you say that 3 is the yellow spot? And if you chose 3, you probably know why. Good. The yellow spot is the area with the clearest vision because of a high concentration of cones. That's perfect, right? Okay, so it's for 1 mark and it's just saying give a reason, right? So either one of that would be fine. If it was explain your answer, then it would have been for 2 marks to say that it's the area of the clearest vision. You can see the vision here on the y-axis. It's the highest point. And then why is that the case? Because we know that the yellow spot has the highest concentration of cones. Name the part of B in the diagram which corresponds with number 2 on the graph. That's for 2.5. So which part of B, which part of the retina corresponds with number 2? Just name. The question is saying name, so you don't need to write the sentence. Perfect. Blind spot, right? And if they ask why, because you can see there's no vision at number 2. It's 0. It's touching the x-axis, meaning it's 0 on the y-axis, right? So there's no vision there. Why? Because that's the blind spot, and at the blind spot we know that there are no rods or cones, so there'll be no conversion of the stimulus into an impulse. Okay, so we go to question 3 then. They're talking of betta Chinese or Siamese fighting fish. That's a very beautiful fish. Similar to humans, the males are very beautiful and the females are more like a bland color. If we look at this, they're saying that the tail color can be blue, which is dominant because they put in a capital letter, or black, which is a small letter, meaning it would be recessive. And then the pectoral fin, we probably haven't heard of that before, but they're giving you a diagram. They're showing you the tail has a color, and then pectoral fin is this fin on the side of the gills. It can either be short or it can be long. Short with a capital F, that already tells you that's dominant, and long would be small f, which is recessive. Then they're telling you that a male fish, heterozygous for both characteristics, is mated with a female that is homozygous dominant for tail color and with a long fin. So if they're telling you that the male fish is heterozygous for both, that would basically be giving you the genotype. Hetero means different. So basically they're telling you it's for the tail color, it's going to be capital B and a small b, which is different. Not a repetition of the same type. And the fin would be capital F and small f, right? Because that's heterozygous. So that would be the genotype of that one. And it's mated with a female which is homozygous dominant for tail color. Homozygous means the same. Dominant would be capital letter. So this would be then represented as capital letter for dominant, homozygous capital letter again. And for the tail color and with a long fin. Now, how would we represent a long fin? We go and look at the table. Long fin is a small letter f. So how would we represent long fin? Would it just be like that? Capital B, and a small f. Do you all agree? No, then what should it be? Should be capital F and small f. Small f, perfect, right? Remember, this is a recessive trait. The long fin here is representing a recessive trait. A recessive trait can only show in the phenotype if it is homozygous recessive. So this had to be small f. Right, then we get to the questions themselves. Write down the phenotype of the male fish. We've got this from the question, which is the genotype. So if you convert that to phenotype, capital B, small b, we've already got the answer there, Humera, thank you, which is capital B is dominant and will overpower the small b, and therefore you're going to get a blue color. And for the fin, short is dominant. Even though you've got short and a long allele, the dominant one will show. So the answer would be blue and short, right? A blue tail color and a short fin. Then write down all of the possible genotypes of the gametes that can be produced by the female. Now we said the easiest way to do this would be to make a Punnett square. And then you take the bees and put them on one side. and then you put the other letter on the other side, and then you join them up to get your gametes, right? So in this case, because it's homozygous for capital B and because it's homozygous for small f, it can only give its offspring either a capital B, it can only give a capital B and a small f. There's nothing else that it has to offer. So the only answer there would be capital B and small f. There's no other possibility there. Then 3.1.3, name the type of cross shown above. What type of cross is this? We've done many types of cross. There's complete dominance, incomplete dominance, co-dominance, sex-linked, blood groups, dihybrid. Good. Right. So this one here, you can see they're talking about two different characteristics at the same time. So they're talking of the tail color and the size of the fin. So since you're looking at two characteristics at the same time, that is known as a dihybrid cross. And then for 3.2, draw a label diagram of a sensory neuron. Okay, so we look at the memo for that one, right? So here we said blue tail and short fin, it was 2 marks. You can see the 2 ticks are right at the end, meaning that if you had blue tail and long fin, you get no marks for that. It's not separated, 1 mark for blue tail and 1 mark for short fin. It's 2 marks all or nothing. The only gamete was capital B and small f. If you had more than one that you wrote, only the first answer would have been marked. And it's a dihybrid cross. When you draw a sensory neuron, it should look something like this here. Okay, so you should have the cell body in the middle, not in the start like in a motor neuron. You'd have, ideally, you'd want to show maybe the direction of the impulse. So if the impulse is moving from left to right, this side would be called the dendrite. And then after the cell body, you label that as being the axon. And you'll have the nucleus within the cell body surrounded by a cytoplasm, and you'd label the different parts. Okay, and you'd also, 1 mark is for the caption, for writing that this is a sensory neuron. So if you drew it perfectly and labeled it without naming the diagram or labeling the diagram itself, you lose 1 mark. Correct representation, did it look like a sensory neuron and not a motor neuron? That's your other mark. And any 3 correct labels of these. Remember, the others are not in the exam guideline. There'll be no marks for that. Those node of Renvier and Schwann cell nucleus, those things are not in the syllabus. So if you label those kind of things, you end up losing out on marks there. Okay, question 3.3. Describe how a high level of thyroxin is brought back to normal by homeostatic control. And we've learned in homeostasis, we said some of the important things is know your control center and know your target organ. What's the control center for thyroxin level? Who can tell us what's the control center for the thyroxin level? Right, for all of those various levels that we speak about, you need to know the control center. Very good. Yeah, it's pituitary gland. And the target organ, we said for whether it's carbon dioxide, for water level, for salt level, for glucose level, know your control center and know your target organ. So very good. The pituitary gland is the control center and the thyroid gland would be your target organ. So describe how a high level of thyroxin is brought back to normal. Your starting point would be to say the level of thyroxin is high and above normal. The control center, which is the pituitary gland, will be stimulated. What will the pituitary gland then do? How will it communicate with the thyroid gland? That's what we need to say now, right? So very good. The response of the pituitary gland will be that less TSH is secreted. Okay, so less thyroid stimulating hormone is secreted. And if there's less thyroid stimulating hormone secreted into the blood, that will go to the target organ, which is the thyroid gland. And what will be the response of the thyroid gland then? If there's less TSH, it already tells you there's less thyroid stimulating hormone, then the thyroid gland will be stimulated less, and therefore the thyroid gland will, good, less thyroxin will be secreted by the thyroid gland. Very good. Okay, so there's your 4 marks there. Picked up by the pituitary gland, which is the control center, which then secretes less TSH. Remember, with the hormones, generally, you're going to be saying less or more always when you talk about the secretion there. And this is sent to the thyroid gland, which is the one that's going to respond the target organ, and it will inhibit the secretion or it will result in less thyroxin being secreted. Okay, so we get now to the last question, which is the essay. Name the structures of the ear that play a role in balance and explain their functioning. Okay, so firstly, let's name the parts in balance. There's certain receptors. What are the receptors? And then where are those receptors found? And what are the stimuli that they pick up? That would be the first part, right, of the structure. So for balance, we've got the cristae and the macula being the two receptors. So let's go with them one at a time. The cristae receptor, or the crista plural, where are they found in the ear? Good. Crista are found in the semicircular canal. Very good. And what is the stimulus that it picks up? So the crista, you'd get a mark for that. Semicircular canal, probably another mark. What is the good? The speed and the direction of movement is picked up. Good. Right. So they pick up the stimulus of speed and direction of movement. Then when we spoke about the macula, you've named that. It's a mark already for naming the receptor, which is found where? Where do we find the macula? Macula are found in the seculus and utriculus. Good. And from the seculus and utriculus, they're going to be picking up which stimulus? The maculae pick up which stimuli? The position of the head. It's best to say position of the head relative to gravity, right? But perfect, good. So we've said that we've spoken about both receptors. We've spoken about where they found and what stimuli they pick up. Then what happens from there? The role that they play, explain their functioning. How do they function? What do receptors do? All receptors do the same thing. Receptors, any receptor in your body will could receive the stimuli and convert them to impulse. Right? So the cristae will pick up the speed and direction of movement, convert that to an impulse, and the macula will pick up the position of the head relative to gravity and convert that to an impulse. And then what happens to the impulse? Good. The impulse is then transported along the auditory nerve, and from the auditory nerve, it goes to the cerebellum, right? Just be careful. It's not the auditory canal, right? Auditory canal, remember, is after your pinna, you've got the auditory canal, which is going to take the sound waves to the tympanic membrane. Right? So it's a similar word. In one word you change, it changes the entire meaning of your whole sentence. You end up losing a mark, not just for the word, but for the whole concept there, right? So the impulses are carried by the auditory nerve to the cerebellum. Remember, balance goes to the bellum, right? Cerebellum. And the cerebellum, does it end there, the story? There's something else that you can add on. Okay, good. And the cerebellum interprets them and sends impulses to the muscles, normally of the legs, and they will restore your balance. Very good. Okay, so the cristae, one mark, name of receptor, found in the ampullae, which is the swellings at the base of the semicircular canals. That's the second mark. Are stimulated by a change in the direction and speed of movement. That's the stimulus. And the maculae, name of receptor, in the utriculus and seculus, which is the position of the inner ear or part of the inner ear, are stimulated by change in the position of the head. Okay, and then they generate impulses, and these travel on the auditory nerve until they reach the cerebellum, and then the cerebellum will then send impulses back to the muscles, and it enables a person to maintain or restore balance and body position. Now, if you look here, for the first part, between the cristae, the semicircular canals, which are in the semicircular canals, and the name of the stimulus, change in speed and direction of the movement. And then the maculae, which is the receptor, the position in the inner ear, and the name of the stimulus. For those six, This memo is saying any 4. So even if you had all of them, you could get a maximum of 4 marks there. The other 4 marks was speaking about the functioning. But this last bullet is in bold and it's having this asterisk, which is saying it's compulsory. You had to end up with this, because if you look at the question, it said, explain their functioning. Their functioning is to maintain balance. So in your answer, you had to have maintaining balance or restoring balance in that process. Okay, that was a very, very important point. Without that, you couldn't get full marks, even if you had for these other 4 points here for functioning. Right? So impulses were generated, 1 mark, go on the auditory nerve, another mark, to the cerebellum, and then sends impulses to the muscles. Any 3 of these 4 you're supposed to have, and you had to have compulsory that they restore balance and body position. Okay, and aside from the 8 marks for content, in this case, there's 2 marks for synthesis. 1 is for relevance. Was everything on topic, basically? Nothing going off the topic. If you spoke about hearing, you'd lose a mark. If you spoke about the auditory canal by mistake, that's part of hearing, you could lose the mark there also for relevance. And then comprehensive, did you answer the entire question? At least 3 out of 4 for the structures, and at least 3 out of 4 for the functions. Right, and that's the end of that paper.